Okay. Hi there. Thank you very much for the introduction. I'm really sorry not to be able to be with you in person, and I should start by congratulating Professor Long on collecting a really terrific program and an excellent group of people to interact with. I just wish I could be there. So I'm going to start by introducing work that's been done in my lab over the past 5 years, together with a set of collaborators, and I think you should be able to see the slides. I work in an institution called SynthSys, which is Edinburgh's Centre for Synthetic and Systems Biology. Our work's been funded by EPSRC, BBSRC, MRC, and the European Union, and some of what I'll talk about today is related to work of a new network in the UK called Multiscale Biology. So this is for modelers working on many different organisms who are interested in multi-scale interactions. So before I go to the first proper slide, can I just check that you're hearing me and that you can see the slides? Do I see any nodding? Yes, I see nodding. That's great. Okay, thank you. So my background is in genetics. I was trained as an empiricist, and you can see that by the people in my past lab list who are left justified. So those are all the ones who are experimentalists. And here at some point theoreticians arrive, and now we have a series of people who are neither on the right nor on the left, but somewhere in the middle, because they do a bit of experimental work and also modeling. For this talk, the people that I should particularly credit are Yin-Hoon Chu and Daniel Seaton, working with a large set of collaborators, particularly on the UK Robust project and the EU TIMET project, including Mark Stitt, who's with you, and Argyris Zardilis, who is a graduate student in the lab who's with you also. The talk's going to cover an example which is one of several different proofs of concept for the plants in silico idea. It's an example of understanding plant growth from the middle out, and in this case we start by modeling a gene circuit, understanding its outputs, creating what we call the framework model for Arabidopsis growth, and I'll describe the process of model integration that led to that framework model. And I'll end then by some thoughts about the community and infrastructure, which we may need in order to build further examples like these and link them more broadly in line with the ambitions of the plants in silico concept. So the biology we're starting with is the biological clock, the 24-hour timer that gives some of you jet lag at the moment, I would imagine, but also in plants drives rhythms in 30% of all genes expressed around the day. So here's a summary of a microarray experiment. These are samples taken from plants in constant light, but you can see there are sets of genes here, hundreds of genes expressed in the evening, hundreds the next morning, through the day, and so forth. Here are two Arabidopsis seedlings growing in those same conditions, in constant light and constant temperature. But you can probably see their growth is not constant, give or take some video buffering. They grow and stop every day. And that's due to rhythmic control by the 24-hour circadian clock. So we've worked almost exclusively here in Arabidopsis. Here it is growing on the sidewalk. There's a Coke bottle cap for scale. You can tell it's a real high-class organism. In Arabidopsis, just as in other plants, the circadian clock doesn't just control elongation. This pervasive control of gene expression also affects many other physiological processes. For example, the day-length control of flowering time is a product of rhythmic circadian regulation. So plants growing in long days in summer flower quickly, and plants growing in short winter days flower slowly, in part because of rhythmic gene expression. So the clock is a useful multi-scale regulator. It controls processes from the intracellular to the whole plant at timescales from the biochemical through the daily to the seasonal. Thanks to the work of a very large number of labs in Arabidopsis, we understand the key components that drive that rhythmic gene expression. They are themselves a circuit of genes expressed throughout the day in a light-dark cycle. This graph shows you data from Mark Stitt's lab, in which we're tracing the RNA expression levels of 2 morning clock genes here in red, down in the evening, up in the morning, the day genes here in yellow, the evening genes in blue. These are plant-specific transcriptional regulators, which we now know are linked together in a complex negative feedback circuit. How do we know that? Again, from the work of many different labs using molecular genetics, mutants, light and temperature treatments, which justify each of these connections in the model. So, over several years, my group, working with other collaborators, have built a differential equation model that represents the function of these genes within the clock gene circuit. The P2011 version has order of 25 differential equations, more than 100 parameters, and those equations represent the activity of genes, their proteins, protein complexes, and light inputs. You may ask whether the model is sufficiently constrained, given that large parameter number, and I can tell you that it is, because the model matches fine details of the dynamic behavior in the circuit. So we have a model, here we're showing 3 components, the morning genes in red again, one of the day genes here in orange, and an evening gene in blue. The time series data has data points and the simulation doesn't have any data points. But you can see we have a good match between the gene expression patterns and the data for a wild-type plant. If we now mutate those day genes, so we're going to remove 2 of them, the first 2 including the one previously plotted in orange, now we can see a change in the dynamics of the gene expression. So this orange gene previously turned off the red one. Now that orange one's gone, the red one takes much longer to fall. Previously it was down by 8 hours, now still up at, still detectable at 12 hours. This is a slow clock mutant. Everything takes much longer to operate in this genetic background. And I'll come to the consequences of that in later slides. I'm arguing here from these data that the differential equation model of our intracellular clock circuit works well for Arabidopsis. So what of other plant species? What do we know about these same genes across crop species, for example? Well, here I'm plotting some of the homologs of the Arabidopsis clock genes that are known in crop species. The gene that I mutated on the previous slide is this PRR7, and that is the homolog of the barley major photoperiod control locus, which is used in breeding, so PPDH1 or PPD-D1A in wheat. Over here, one of the evening genes, L3, I'd highlight. This is MAT-A of barley, or the HD17 flowering time QTL in rice. So these same genes that control the clock in Arabidopsis also control photoperiodic flowering in crop species. A paper last year showed that one of the input gene homologs in tomato is mutated in all cultivated tomatoes, such that the tomato clock also has a slower pace than the 24 hours that you might expect. But in that case, it affects vegetative phenotypes they claim. So we have a gene circuit that controls flowering in Arabidopsis, and we know those genes are important also in crop species. How can we understand how they affect flowering? Well, the first way to do so is to go back to Arabidopsis and ask what the links are between the clock circuit and the flowering time machinery. And that's what we did under Karen Halliday's leadership in the Robust project. So we're asking how the clock connects to flowering, but also to elongation. To cut a long story short, those connections are more of the same. A transcriptional network in which clock transcription factors regulate a number of intermediate proteins. We now, in 2015, published a model with all of this output machinery. It built on some earlier models, which we published previously. One innovation that we made there was that Daniel Seaton was able to connect this pathway to genome-wide targets. So our model of the clock now controls not only these physiological targets, but also hundreds of genes across the genome, matching microarray data which were previously published. So in that case, we were doing more transcriptional network modeling. Our challenge in the EU TIMET project was to go further than the transcriptional networks, to ask how the clock controls plant growth. In order to do so, we needed to understand how the clock controls at least some key aspects of metabolism. And the aspect of metabolism TIMET focused on is the control of starch degradation. We had two requirements in that project. One was to understand more about the clock control of starch, which Alison Smith and Mark Stitt conducted experimental work to do. But also we needed a growth model. If we're going to connect the clock to growth, then we need a model context to do so. And that's perhaps the most relevant part in the context of plants in silico. No such model previously existed, and we chose to build it in a style that would be quite familiar to crop modelers. So our first question was then, could we construct a growth model for Arabidopsis using pre-existing components? We were aware of several potential component models which had been published in the literature, and the question was whether we could combine those to produce an accurate model of Arabidopsis growth. And then, secondly, could we use that model to understand the clock's effect? So the first question we addressed using what I'll now introduce as the framework model. So the Arabidopsis framework model combines these four previously published components. I'll go through it for a couple of minutes on this rather complicated slide. So the first model is a simple model of leaf carbohydrate metabolism constructed in differential equations, which, for example, includes photosynthesis, respiration, and the sugar-starch partitioning. The second component is a functional structural model, again, a very, very simple representation, but which includes source-sink partitioning, so that the sugar produced from photosynthesis in this model is now divided among the different organs of the plant. This produces a simple representation of plant structure, including, crucially, the rosette area, and that rosette area is then provided to the carbohydrate model to power photosynthesis. The more area, the more photosynthesis. Here's our positive feedback. The third component was a photoperiod flowering model, including a clock gene expression circuit and the control of the key flowering regulator FT. And the last component is a multi-factor photothermal model that includes vernalization, a thermal time component, and together this model ends the simulation. So at flowering, the simulation stops. The framework model, therefore, is just really a model of Arabidopsis rosette growth. We also have a root compartment, but we don't go into the inflorescence. These are the building blocks of the model. Having building blocks is not a guarantee that when you put them together, your edifice will stand up or will accurately represent the data that you can produce. These four models had never previously seen each other. They were constructed completely independently on in different countries, in some cases different continents. A crucial question, therefore, was whether their outputs were sufficiently compatible not only to match their data reciprocally, but also to match our data from Edinburgh. Yin-Hoon Chu in the lab demonstrated that that was the case. So here we have a little more complex representation of the model. I'd like to draw your attention to the data at the bottom. Here we have data points for the growth of the whole shoot or for the mass of each individual leaf throughout development. And you can see that the model simulations in green closely match the data points in black. This was published in PNAS last year. So, at least for these reference conditions, we have successfully combined models without modifying their parameters. So of 126 parameters in total in this model, we changed two, one of which was the water content, and the other of which was this flowering threshold. So flowering really is variable among labs. Nonetheless, this suggests that it should be possible for other groups working on Arabidopsis to contribute further components to a model of this type. So that was the question about whether we can construct a model. The second question is whether we can now use that model in order to understand how the clock affects growth. To do this, we've produced a second unpublished version of the framework model in which we now include the whole of that complex clock model and its outputs to hypocotyl elongation and flowering time. This clock model controls starch degradation as well as controlling FT, and through starch it may therefore control growth. So, does it do so accurately? The first requirement to understand that question was to characterize our slow clock mutant in much greater detail. These are data coming from Mark Stitt's group, and he was one of the groups within the time at Consortium who contributed to that characterization. In this panel here, you can see the starch profile for our slow clock mutant. Its slow clock is slow to predict dawn, and so starch degradation is timed not to run out at dawn, as in the wild type, but it's 4 hours late. So you can see the red line here. Starch is being degraded for a target of about 28 hours instead of 24. And that is nicely matched by our slow clock model, as I'll show you. The first question, though, is whether we can use the framework model to address our clock effects. So, if we alter starch degradation, can the framework model accurately predict biomass? In this case, we've tested that very simply using a mutation called LSF1, which directly affects starch. The LSF1 mutant accumulates a high level of starch, it fails to use it effectively to grow, and therefore its biomass here is reduced compared to the wild type. You can see in the photos of the plants, that's a subtle effect. LSF1 is really rather slightly smaller than Columbia, and this is the clock mutant we're now trying to understand. So the question is now, if instead of mutating starch degradation directly in the model, we rather control starch indirectly by mutating the clock sequence, does the model now represent biomass accurately? So within the framework model, we've now replicated that slow clock mutant only in the clock component. That alters starch degradation such that the mutant has a higher starch level, though not quite as high as the LSF1. You can see in the middle panel that biomass growth is indeed reduced in the slow clock mutant. And this is the effect of the starch defect is shown in the black line and inset here. So the clock mutant plant has a biomass of about 0.55 grams. And our black simulation here is predicting about 0.65. So we can say, therefore, that the starch mechanism reduces biomass, is predicted to reduce biomass by nearly as much as we see in biology, but not by quite the full amount. So that argues that starch is the major mechanism through which the clock can regulate biomass growth in Arabidopsis, but that there may also be a minor mechanism. We have some good ideas as to what that is, but in the interest of time, I won't go through that. This example then shows how it's been possible for us to reuse existing models and to connect an intracellular gene circuit through a set of quantitative mechanistic models up to the scale of a whole plant biomass. Our next steps are to go from this middle level back to the genome. I showed you one example of this, and another student in the lab is now working on the binding site sequences for some of these transcription factors. We also want to extend the model around the life cycle to model inflorescence and seed production. I mentioned, though, that the model had been constructed intentionally in a modular fashion so that we could swap out some parts and replace them. The Stelling and Gruesome AT cell model, which you'll hear about tomorrow, would be one candidate as an example where we could replace our very simple representation of metabolism with something more complicated. Or Professor Long and Jinghan Zhu's e-photosynthesis model might replace our extremely simple representation of photosynthesis. In the context of plants in silico, you could say that what we've done so far is really just to link genotype to whole plant phenotype. That's not a bad step. But Plants in Silico aims, I think, to go much further than that. I mentioned that we want to start from the genome, then to connect genotype to phenotype in a particular environment, but not in a growth chamber. We want to understand the connection to field traits. If we can make that connection, then for a particular management, we may be able to understand the components of yield, again, starting from this low mechanistic level. If we include understanding of genetic variation, we can then understand past effects of selection and how we got to the genome sequences, for this clock circuit at least, that we started from. Some of our experiences in running part of that circuit may be instructive for the roadmap as a whole. So I should say very clearly, we came from the modelling tradition in the field that calls itself systems biology. And in systems biology, we've had 10 or 15 years worth of building infrastructure, including a model representation called SBML, which was set up to exchange models between software tools. Because there's a standard model representation language, we have a database called Biomodels in which hundreds of systems biology models are available. And again, it's possible to produce software tools which produce or consume SBML. So our clock and metabolic differential equation models fitted very naturally into that whole framework, and the functional structural model was simple enough that it also would have fitted into SBML. However, we have two problems. Like all crop models almost, our model reads in weather data. Now, no systems biology model is used to doing that. And secondly, we dynamically create new compartments in the model, which are leaves. Of course, this is entirely standard for plant growth, but again, completely different from most systems biology models. So, being out with the SBML representation, we therefore lost all the software tools that we might have used to disseminate our models. And we couldn't, the components that we had put together in the framework model were widely scattered. In fact, for one model, the only way that we got the file was to search it, scrape it back out of the Google cache. So some of those models were at severe risk of being lost entirely. In response to that, we produced a model repository called the Plant Systems Modeling Repository, or PLASMO. It contains, originally started with just a dozen models. We refactored them from whatever programming language they started in and represented them in an XML format which we felt would be easy to convert. And it's also rather similar to SBML. Some of the models in Plasmo are indeed SBML models. If you wanted, for example, to access the first version of the framework model, you can go to our website, there's the address, get model number 76. You have all of the files that you need, including MATLAB versions and the versions that would run it on your desktop in a commercial package called Simile. Now, we wouldn't have gone to a commercial package except that we'd lost all the SBML tools. And Simile does represent the model components that we needed. So, after downloading your files, you can upload them into Simile. It produces this really rather nice graphical representation of the model, except that the more complicated the model becomes, the harder it is to use a graphical interface. You can then edit any aspect of the model and run it. However, we discover, of course, that we are bridging between very different communities. Systems biology, Arabidopsis on the one hand, crop science on the other. And the experience is always that these communities have very different practices. So for plants in silico, I'd suggest that Arabidopsis will continue to be crucial for the data density and for the biological tools that it provides. But I think that our simile representation was hardly used at all in the Arabidopsis community. It was really too complicated for them to use, despite the fact that it had graphical interface and so on and so forth. So for version 2, we've taken a very different approach, and we've just provided an online simulator in which you can change very little, but it runs the model for you right in your web browser. It's active now. If you want to try that web address, you can run the model anytime you fancy. But you could only change the environmental conditions. For plants in silico, aspects like these will be crucial to develop trust among the different community participants that is then going to allow sharing. As a great example of that, I'd like to highlight the AgMIP project, where they really are bringing together models that have not previously been compared. I'd note that the dynamics we suffered from in the crop science models, where models were being lost because their originators were retiring, in the next 10 years, that's going to happen in plant molecular biology. And of course, you'll be well aware of the technical aims that a roadmap will have to address: benchmarking, linking across multiple models like the virtual organisms have already done, and deciding on the best model formats, both for simulation and also just for understanding the contents of a complicated model. So I commend those and the other challenges which you'll be well aware of to the rest of the workshop, and I look forward to discussing some of those with you in the later sessions. Thank you.